Welcome to this presentation featuring non-human primate research models in gene and cell therapy with a focus on potential germline integration. My name is Julie Forget and I'm the Senior Director of Safety Assessment at Altasciences. Co-presenting with me, my colleague Kayleen Koenig, Associate Director of Molecular Biology and in charge of the PCR department at Altasciences. On today's agenda, we will briefly review key study design items from a regulatory perspective, outline the specialized tissue collection strategies employed at our facility, and describe the downstream sample processing and analytical workflows used to isolate DNA and RNA for subsequent analysis. As we know, ICH S12 provides valuable guidance and insights relevant to study design for gene therapy programs. With respect to selection of a biologically relevant species, non-human primates are frequently used in gene therapy studies due to their close similarities to humans across genetic, physiological, immunological, and developmental domains. From a safety perspective, and within the scope of today's presentation, there's a focus on evaluating the potential for random integration into host genomic DNA. This assessment is closely linked to the need to characterize biodistribution in both target and non-target tissues, as well as in relevant biological fluids. Ultimately, the success of these assessments is highly dependent on how tissues are collected, handled, and preserved. Tissue collection strategy is therefore a critical component of study design and execution. With that, I'll hand it over to my colleague Kayleen, who will walk us through several key aspects and best practices related to tissue collection.
Thank you, Julie. Biodistribution assessments are to determine where the therapeutic ends up in vivo. So we sample anywhere between 13 to 25 tissues per animal, ideally the target tissue in addition to non-target tissues. A subset of the FDA recommended standard list, as well as some expanded panel tissues, are listed on the right side of the slide. As downstream DNA or RNA analyses rely on properly collected samples, we are able to implement meticulous collection procedures at Alta for the collection of all those tissues collected at necropsy. To take these general collections a step further, we have established procedures to minimize cross-contamination between tissues. Some of the implementations include using new disposable scalpels to make tissue cuts, or the use of disinfected or sterile instruments. We also have surface cleaning procedures, for example, when, say, whole organs are removed and weighed before being sectioned for the sample collection. And when the study context is outlined, we can order the tissue collections when certain target organs or notable target organs or tissues may be warranted to be collected before other informative tissues. And when samples are collected, we can employ the use of preservatives or implement rapid storage procedures when needed so that DNA and RNA within a sample remain intact for whatever the downstream analysis may be. Part of this collection also involves the harvesting of the gonads, which are collected for a number of different data endpoints, including histopathology and biodistribution, but also for downstream gamete analysis.
Improper isolation of gametes has the potential to introduce false positive results, mainly due to contamination with somatic cells. For this reason, it's critical that strict and well-controlled protocols are followed throughout isolation, purification, and cryopreservation to preserve cellular integrity and ensure suitability for downstream nucleic acid analysis. In males, gamete collection is performed prior to study termination and requires the use of sexually mature animals. Semen samples are collected and mixed with a cryopreservation buffer, followed by controlled freezing and storage in liquid nitrogen. Prior to nucleic acid isolation, samples undergo a series of sequential washing and lysing steps designed to remove contaminating somatic cells while preserving sperm integrity. Once this purification process is complete, the resulting intact sperm heads are suitable for DNA and RNA isolation. Female gamete collection presents a distinct set of challenges due to the small size of the ovaries and the fact that oocytes are embedded within ovarian tissue rather than existing as freely isolatable cells. In addition, the limited amount of tissue available for assessment of potential germline integration must be carefully balanced against other critical toxicology study endpoints that cannot be compromised for regulatory or scientific reasons. To address these constraints, while maintaining the integrity of all required endpoints, our laboratory has successfully implemented a balanced tissue allocation strategy that allows comprehensive safety and biodistribution assessment. One ovary is dedicated to histopathological evaluation, ensuring robust morphological and safety assessment. The remaining ovary is divided with one half allocated to biodistribution analysis and the other half cryopreserved to allow for potential future germline or molecular investigations. This approach has proven effective in meeting both regulatory expectations and study objectives without compromising data quality. Cryopreserved ovarian tissues can be retained for extended periods and remain suitable for future gamete isolation and analysis for several months. Based on regulatory feedback, oocyte isolation does not always need to be performed immediately and may be conducted either ahead of submission or post-submission should a concern regarding potential genomic integration arise. This flexible approach allows programs to balance study scope, regulatory expectations, and maintain study timelines. Oocyte isolation is a highly specialized procedure that must be performed by trained and experienced personnel. The process involves the careful manual denudation of each oocyte, one at a time, to completely remove surrounding somatic cells. This step is critical as residual somatic cell contamination could interfere with downstream molecular analysis and lead to false positive results. Once gamete isolation is successfully completed, the purified samples are transferred to the appropriate lab for processing and analysis of DNA and RNA.
Now I would like to go over some of the practical data which we have collected internally, which has allowed us to perfect our gamete processing techniques in non-human primate samples with regard to obtaining DNA and RNA for further analysis. After successful collection of denuded oocytes and washed and isolated sperm cells, as Julie previously described, we were able to successfully isolate DNA from both sample types, which was able to be amplified in library preparation for next generation sequencing, or NGS as I might refer to it. So to reiterate the variables that we incorporated at necropsy, half of an ovary was collected and slow frozen for more than 3 months. And from these half ovaries, the team was able to collect anywhere from between 21 to 65 oocytes. For oocyte DNA isolation, we used a ChIAMP DNA microkit with a mini elute column, and we eluted 40 microliters, which was sufficient for library preparation for downstream NGS. We do want to note that the DNA concentrations for both sample types here was determined using a nanodrop spectrophotometer. And so regarding the oocyte DNA specifically, we do acknowledge that this is a notable overestimation. So realistically, another quantification method is required for more accurate DNA concentration determination. 1 milliliter of semen was utilized and washed for sperm cell isolation to remove the somatic fraction. And then samples were collected from our colony animals. They were stored for at least 2 months in frozen storage. Sperm counts ranged from 1.5e to the 8 to 3.5e to the 8. So after washing, the QIAGEN DNase blood and tissue kit was used, and we also eluted in 40 microliters. NGS analysis was successful, and we were able to confirm that the DNA samples we isolated from the oocytes in the sperm were able to be analyzed. This slide is showing our RNA isolation results from gametes that we isolated. So the data shown here, there were a few different variables involved compared to those of the gametes for the DNA isolation. The oocyte counts are very different. They range from 16 to 126 oocytes, and these were from whole ovaries. So for the DNA isolation data on the previous slide, those oocytes were isolated from half ovaries. For the gametes that we used in RNA isolation, they were, again, whole ovaries. Some of these ovaries were also stored at -80 for as long as 6 months, so it was a much longer storage period as well. We used the RNase mini isolation kit to isolate RNA, and concentrations were again determined using nanodrop and are also approximate. So we acknowledge the detection limitations of this instrument. For the semen processing and sperm RNA isolations, we were able to use the same wash steps for removing the somatic cell fraction from either 0.5 or 1 mil of cryopreserved semen. So I do believe these were not in storage as long, though, so likely about a month. These all came from our stock colony. So as we would expect, the RNA concentrations from the sperm are much lower than those of the DNA from sperm, but concentrations were slightly higher due to the sheer amount of sperm cells compared to the individual oocyte cell count. So that's just a comparison to be made aware of. And here are some example applications which can be applied once there are DNA or RNA isolated. So we've got short read sequencing, which is likely the most affordable and routinely employed by our sponsors. There is single cell sequencing available of either DNA or RNA, and that would be much more informative for the respective therapeutic for directly assessing integration, but it also can be rather costly. There is also Sperm-seq, which caters more toward having an increased number of cells to sequence. Of course, there are qPCR and dPCR techniques, either for intermediate library prep or target quantification, depending on the context of the study and any biodistribution data that was previously obtained. So there's a couple different options here. And to close us out, Julie and I would like to thank you for attending our talk here today, and thank you so much.